All right, we are looking at a cell, and the extracellular space is up above. This is the bloodstream, and you can see that you've got some glucose dissolved in the blood, and that would make this environment right here the cytoplasm. So when you consume a meal that contains glucose, your blood sugar levels rise. That is going to trigger the release of insulin, this small peptide hormone, so it's a protein, and it's going to dissolve in your blood and it's going to travel to target cells. A target cell is just a cell that contains a protein receptor that's capable of binding to the signal molecule. So this could be like a muscle cell, it could be like a liver cell. The point is that it's a target cell because it has an insulin receptor. In purple. So insulin is going to bind to the receptor, very similar to how an enzyme binds to substrate. Now the receptor isn't catalyzing a reaction with insulin. It's not like breaking it down, or it's not doing something to the insulin. It's simply binding to the insulin, and it's going to send a signal to the cell. The receptor will send a signal to the cell once it binds with insulin, and the exact mechanisms of that we'll talk about next unit, but it's going to send a signal to the cell, and as a response, the cell is going to send a vesicle containing GLUT to the membrane. So the green square is GLUT, which is a glucose transporter protein. Once the vesicle fuses with the membrane, notice the position of GLUT. It's positioned in the vesicle in such a way that it's not going to be released into the blood. Instead, it's going to become incorporated into the actual plasma membrane. So here's your GLUT integral protein. Now, glucose has a pathway to flow into the cytoplasm. The glucose concentration in the blood is high relative to the glucose concentration in the cytoplasm, and so diffusion is going to occur, and glucose will passively move, passively diffuse across the membrane, thanks to GLUT, into the cytoplasm. So this is how glucose gets into a cell from the blood. And the first thing that's going to happen is glucose is going to be phosphorylated. Glucose is going to be phosphorylated. So a kinase, it's called hexokinase, a kinase enzyme is going to take glucose as a substrate. It's going to take ATP as a substrate. And it's going to rip a phosphate off of ATP, and it's going to transfer it onto glucose. This accomplishes two things. So let's rip this phosphate off and transfer it onto glucose. Okay, so this accomplishes two things. The first thing is that it helps keep the concentration of glucose in the cytoplasm low. So this ensures that glucose continuously floods in as long as there's a pathway available. The second thing it does is that it makes this glucose molecule unreactive. It makes it unable to leave the cell. It makes it highly reactive. Sorry, I said the opposite. It makes it highly reactive. Remember, anytime we add a phosphate onto something, we make it more unstable. And so this instability is going to help split glucose in a process called glycolysis. But that's the reason for phosphorylating it in the first place. Keep it trapped in the cell and make the glucose concentration low so that glucose continues to flood in, and it makes it reactive. At this point, the cell has an important sort of enzyme-mediated pathway that it needs to regulate. And we've discussed this before, but at this point, the cell needs to choose whether or not to send it to a synthesis pathway or a synthetic pathway. In other words, build this glucose up into glycogen for long-term energy storage, or, I keep losing my mouse, or are we going to go down the breaking down pathway? Is it time to destroy glucose in order to extract those high energy electrons from it and use that energy to produce ATP. And so there are a couple enzymes here that need to be regulated through allosteric regulation mechanisms, through feedback mechanisms, in order to know, or in order to determine the outcome or the fate of this glucose molecule. And so in the next video, we're going to go down the breaking down fate